Good morning, everyone. It's a tremendous pleasure for me to introduce my former PhD supervisor, Trevor Thorpe. And the session is named after him in honor of Trevor Thorpe. It is the inaugural symposium on that, Trevor Thorpe Symposium. Trevor Thorpe, many of you probably are familiar with him, and he's been a really great leader for plant tissue culture in terms of the discipline, plant tissue culture. Trevor was born in Barbados and then came to North America after a period of study, undergrad years in India. So, which I discovered after I went to his lab, by the way. Anyway, so after coming to North America, he did his PhD under the supervision of Murashige. And the photograph that you see up on the top left is with Skoog and Murashige. Trevor standing there with my senior, Indra, who is sitting in the audience here. So fellow grad student from the lab, right? So Trevor after he completed his PhD in Murashige's lab, he did a couple of years of work at USDA lab in Pasadena before moving to Canada. So from Barbados to Calgary, that was a major or a series of moves. And since then, he stayed on at the University of Calgary. That's basically where I did my PhD in his lab, right? So until he retired there in the year 2000, right? So Trevor has always had a very good mix of people in his lab. So much so, at some point, we would be like 20, 24 people in the lab from 15 different nationalities. And the neighbors would call us the mini United Nations, right? So he would attract talent from all parts of the world, either in the form of students, grad students, visiting scientists, sabbatical visitors from all over in Asia, from Japan, from Korea, from China, from Malaysia, all over, right? So during his long career, he has played extremely active roles in the societies, including this society, and he was, among others, Lifetime Achievement Award winner. Move to the next, right? So the contributions from Trevor, if you go to the pre-internet eras, we used to depend on books and published papers from journals and things like that. So one of the important contributions many people from the older generation would know is the book that I have, a photograph of a plant tissue culture book edited by Trevor. Most people were referring to that. And then he had an extensive series of publications in conifer biotechnology, including the somatic embryogenesis, et cetera, various micropropagation strategies. And his outstanding achievements in physiology and biochemistry of plant tissue culture is something he carried forward, advanced from his study years from Murashige's lab, right? He has served as a wonderful coach for quite a large number of students and postdocs. And in the early years, he was working on the tobacco system. And in the later years, the focus shifted largely towards the conifer biotechnology. And Trevor was a genuine friend of the society and most of the members in the society here, as well as the International Association of Plant Biotechnology and Tissue Culture, or IAPB right now, right? So he was a friend of all those. I've listed a few names there. It is just not an exclusive list. It is just a sampling of it. Murashige, Komomini, Vasil, Gamborg, Erickson, countless other members you can see in some of the photographs that I've shown out there, right? So he had organized the IAPB meeting in 1978 in Calgary. The top photograph is from there. Some of you might recognize people like Olaf Gamborg in there, the younger people, younger versions of the people there. And the bottom left is the 2006 IAPB conference in Beijing. And the bottom right is when Professor Melvin Calvin visited our lab in Calgary. Trevor brought him along to the lab, and then we all took pictures with him, of course. So this is Trevor Thorpe in short for you. We have only fond memories of Trevor now. So a group of his former students and postdocs and associates, as well as friends and colleagues from SIVB as well, contributed to set up a fund as a legacy. So it's a new legacy fund for SIVB Plant Symposium, and we are at the inaugural symposium named after Trevor Thorpe. And those of you who are interested in contributing to the fund, I'm told that SIVB will continue to accept contributions to that, right? So with that, I end my brief introduction for the inaugural symposium named after Trevor Thorpe. Thank you.
Thank you, Dr. Prakash, for such a great introduction to Trevor, and also telling us about the contributions and all the students that came along to SIVB with him. Thank you so much. With that, we are going to begin our session today on plant transformation, advances in plant transformation. Before we start the speeches, I want to ask you, how many of you have been able to develop a plant transformation system in first attempt? Few hand shows. A few, maybe 4, 5 people? Okay. So it is a challenge. There are multiple things that come into play when you're trying to work on a transformation system. And that's a portion of it we intend to cover today. With that, I'm going to invite Dr. Yurong Chang to introduce our first speaker for the session today.
Next slide. Hey, good morning. My name is Yurong Chen. I'm with Bayer. I'm leading transformation discovery with Bayer Crop Science. It is my great pleasure to introduce the first speaker of this session, Dr. Kalen Maisel. Dr. Kalen Maisel currently is a distinguished professor at the Institute of Agricultural Biosciences, Oklahoma State University. And he received his bachelor degree in agriculture from University of Agricultural Sciences in Bangalore, India. A master's degree in horticulture from Clemson University, and a PhD in genetics in Purdue University. And his research centered on molecular plant-microbe interaction, abiotic stress tolerance, and plant genetic resources development. And he has authored over 275 peer-reviewed manuscripts in international journals. And he is also the recipient of Fulbright NAHO Academic and Professional Excellence Award and Ruth Allen Award from the American Phytopathological Society. Karen, floor is yours.
Thank you, Yurong. First of all, I'd like to thank the organizers for giving me an opportunity to present our work. So today I'm going to talk one of the projects in the lab where we have engineered Agrobacterium to improve plant transformation. So before I go there, I just want to remind people that Agrobacterium is actually a pathogen. It's a soil-borne pathogen that causes crown gall disease. And over the past 4 to 5 decades, people have worked on it and tried to understand the mechanism of this crown gall disease, and they discovered that there is an exchange of, there's a transfer of DNA from Agrobacterium to plants that causes the crown gall disease. And this led to improvement of the Agrobacterium so that it can be used for genetic engineering of plants. And this kind of revolutionized the plant biotechnology. Even though there are several ways that DNA can be transferred to plants, even today, Agrobacterium mediated transformation is the most commonly or preferred method used both in industry and academia to make transgenic plants. So we have learned a lot about Agrobacterium mediated transformation in the past 4 to 5 decades. We know how the tDNA is processed from the TI plasmid. We know the proteins that are involved in this process, and it forms the Agrobacterium forms a type 4 secretion system through which the tDNA enters the plant cell along with several other virulence proteins. And these virulence proteins also plays a role in integrating and transferring the tDNA to the nucleus and integrating it. And we still don't know a lot of things about what happens in the plants, even though we know a little bit about it, but we don't completely understand how the tDNA gets transferred and integrated into the chromosome. And my lab is trying to identify the plant components or plant genes involved in this process. So even though we know a lot about Agrobacterium, there are still a lot of bottlenecks in transformation. So one of the main bottleneck is recalcitrance of many crop species for transformation. As you know, not all species can be easily transformed with Agrobacterium. And even if a particular species can be transformed, not all genotypes or varieties can be transformed. So that's a big challenge. And this recalcitrance is mainly because of 2 issues. One is the plant defense responses that is generated by plants against Agrobacterium that can inhibit transformation. And the other one is the inability of the plant to regenerate. So I'm going to focus more on the plant defense response today and touch a little bit on the plant regeneration as well. And another bottleneck in gene editing is that right now that the Cas9 has to be transgenically expressed, even though we heard many talks yesterday about transgene-free editing, but it's still in the development stage. It's not applicable to all plant species. So as of today, we still need to stably express Cas9 in most plants to achieve gene editing. And this can be an issue because this has to be segregated out to avoid non-targeted edits and also for deregulation. And this can be a challenge and takes a lot of time, and especially it's a challenge for trees, tree crops, and also for some vegetatively propagated plants. So I'm going to provide a kind of a solution towards this at the end of my talk. So the plants have a defense mechanism to recognize pathogens and induce a defense mechanism. It's called PAM-triggered immunity. PAM stands for pathogen-associated molecular patterns, and this can be recognized by a receptor called PRR, pathogen recognition receptor. And whenever the plant can recognize these PAMs, it can trigger immunity, that is the PAM-triggered immunity. So Arabidopsis, as you know, it's easy to transform, at least by flower dip. A lot of people do this in their labs. However, Arabidopsis is still not easy to do a conventional transformation. For example, if you want to do a transient transformation, if you want to infiltrate the Agrobacterium into the leaves of Agrobacterium, it's not easy to get the transient transformation. That is because the plants can recognize the Arabidopsis, especially can recognize the Agrobacterium as a pathogen. So the PAM that is recognized is called the EFTU, elongation factor 2. And then the receptor in Arabidopsis is called EFR. So this receptor, this work was done by Cyril Ziffel in 2006. They published very nice work. And they showed that this receptor can recognize EFTU and trigger a plant defense response in Arabidopsis that can lead to reduced transformation. And this EFR is present only in Brassicaceae and not really present in other plant species. So we still don't know what the receptor is in other plant species against Agrobacterium. And we also don't know what kind of PAMP is recognized in other plant species. So that is something still yet to be discovered. So my laboratory also showed a long time ago that not only in Arabidopsis, but also in other plant species, for example, in this case, tobacco, when you induce plant defense response, it can decrease transformation. So what you can see here is that when you increase the salicylic acid, when you spray the salicylic acid on tobacco leaves, so what you're seeing is the tobacco leaf discs transformed with a non-tumorigenic Agrobacterium strain that has a Gus intron gene. You can see that as the salicylic acid concentration increases to the right, the transient transformation decreases. And also we can reduce the salicylic acid in tomato. For example, we can make a transgenic plant in tomato by overexpressing a gene called NAGI, which encodes the salicylate hydroxylase, which can reduce salicylic acid concentration. So when you reduce the salicylic acid in plants, you can see on the left, the NAGI plant is more transformable compared to the control, which is the moneymaker plant that was used for transformation. So not only transient transformation, the plant defense response also plays a role in stable transformation. So here we used the tumorigenic strain that can cause tumors, and we sprayed the Nicotiana benthamiana plants with, and also tomato plants with the BTH, which is a homologue of salicylic acid, to induce plant defense response. So when you induce plant defense response, you can see that the crown gall disease significantly reduced both in Nicotiana benthamiana and tomato. And we also silent some of the genes involved in salicylic acid mediated defense pathway, for example, ICS, NPR1, and SABP2. So when you silence these genes, then you can actually increase tumor formation, which means that it can increase transformation. So all this data clearly shows that the plant defense response plays a negative role in transformation. So even though that the plants produce this defense response against pathogens, for example, in this case, Agrobacterium, the pathogens have evolved to overcome this defense response. And probably even the Agrobacterium can overcome this defense response, especially in naturally infecting plant species. But however, in lab, we always use non-natural hosts. So that's the reason that we see a lot of plant defense response. However, for example, Pseudomonas, it's been well studied how the Pseudomonas can suppress plant defense response. So Pseudomonas injects proteins into plants called effector proteins through type 3 secretion system. And these effector proteins are known to suppress plant defense response. And this interferes with PTI, that is the PAM-triggered immunity. And this leads to effector-triggered susceptibility, or in short known as ETS. So one of the well-studied effector in Pseudomonas is AVR-PTO. So AVR-PTO is known to suppress many different PAMP-induced PTI, for example, even EFR, FLS2, etc. So it has been well studied, and almost a decade ago, FUMI categories lab in the University of Minnesota, they showed that you can actually overexpress AVR-PTO in Arabidopsis and increase the transient expression in Arabidopsis. So even though this is a good strategy, you can have a transgenic plant expressing AVR-PTO so that you can further use that plant for transformation. But the challenge is that AVR-PTO is actually toxic to plants, so you have to use an inducible promoter. And also, you don't want to deal with another transgene, so it can lead to other deregulation issues. So we thought that maybe we can deliver AVR-PTO through a secretion system directly from Agrobacterium into the plant instead of making a transgenic plant. So before that, I just want to give you a brief overview of the secretion system. So Agrobacterium has a type 1 secretion system and type 4 and type 6 secretion system. It does not have a type 3 secretion system. So the DNA and proteins are transferred through type 4 secretion system. And type 6 secretion system is known to transfer some effectors. And it's a newly discovered secretion system, so we don't completely understand the full mechanism of type 6 secretion system yet. So in contrary to this, the pathogens, for example, Pseudomonas has a type 3 secretion system. So we wanted to see if we can engineer Agrobacterium with a type 3 secretion system. Can we put in there and then express AVR-PTO so we can deliver AVR-PTO through Agrobacterium to plant cells. So we also initially thought we could also use the type 4 secretion system. We can fuse AVR-PTOs to some of the type 4 secretion system proteins. However, we thought that maybe we shouldn't compete with tDNA transfer. So that's why we chose to use an extra type 3 secretion system, which does not compete with the type 4 secretion system. So luckily, the type 3 secretion system is encoded by a cluster of genes in Pseudomonas, and that's already been cloned into a plasmid called PLN18. It's a huge plasmid, around more than 50 kb. And these people have already shown that this can work in trans. For example, this can be transferred to other Pseudomonas strains that does not have type 3, and you can make the type 3 functional. So this was already shown before. So we thought that we can use this to see if it works in Agrobacterium. So other thing is that the proteins that comes out of this type 3 secretion system need this type 3 signal. So all of the epitope proteins has this type 3 signal that eventually gets cleaved in the plant. So for this, we first wanted to show that the type 3 or the protein can be secreted through type 3 secretion system. So we used the Agrobacterium strain, non-tumorigenic Agrobacterium strain, where we transferred the PLN18 that has the type 3 secretion system, and we fused the AVR-PTO with a small fluorescent protein called phylau, and we cloned this into a expression vector. And then we kind of performed a Western blot to look at where the protein exists. So if the protein is in the supernatant, that means that the protein is excreted out of the Agrobacterium. If it is still in the pellet, that means that it's not excreted. So as you can see in the last column, the GB2260 that has both AVR-PTO phylau and also the PLN18, you can see that the phylau protein both in the pellet, because not all the proteins will be excreted out. There will be some remaining in the cell. That's why you see in the pellet. And also you see in the supernatant. So if you're seeing in the supernatant, that means that the protein has to be secreted out of the cell, the Agrobacterium cell. So this shows that the type 3 secretion system can be functional in Agrobacterium. To further show that the protein can actually goes into the plant, we used a split GFP system. This was developed by a couple of groups in UC Davis. So in this case, the GFP 1 to 10 is in one fragment, and GFP 11, which is a very small fragment, is split into a different fragment. And then GFP 1 to 10 was transiently expressed in Nicotiana benthamiana, and then we fused the AVR-PTO to GFP11, which is a very small fragment, and then we infiltrated the Nicotiana benthamiana plants. And you can see on the right side bottom square, you can see the fluorescence, which means that the GFP11 is being entered into the plant, and it meets the GFP 1 to 10 that's already expressed in the plant, and it can see the fluorescence. Whereas the controls that has only the GFP 11 or only the PLN18, we do not see any fluorescence. So that clearly shows that the protein can actually be secreted from Agrobacterium into the plant cell. Then, in addition to AVR-PTO, we were also looked at other effectors that can actually suppress PAM-triggered immunity. So we chose HOP AO1 and AVR-PTOB to see if they also can suppress plant defense response. So this is the schematics we did. So for the transient expression system, we used a non-tumorigenic Agrobacterium strain, and we transferred the type 3 secretion system, PLN18, into the Agrobacterium. Then we have a binary vector, which has the Gus intron gene. And then we have another expression vector that has AVR-PTO with its own promoter. For stable transformation, we used two different things. We used a tumorigenic Agrobacterium strain. In this, we transferred again the PLN18, that is the type 3 secretion system, and again the AVR-PTO expressing vector. And also for non-tumorigenic, we also used a non-tumorigenic Agrobacterium strain for stable expression. So in this case, along with the type 3 secretion system and the AVR-PTO expression vector, we also have another binary vector, PCAS1, that has a PPT-resistant gene within its tDNA. So when we did the transient expression in Arabidopsis, you can see that the transient expression, you don't see a very strong GUS expression while just using the regular EHA105, the wild type Agrobacterium strain with the Picambia binary vector. Whereas if you see at the last column, last row, you can see that when AVR, PTO, and PLN18, that is the type 3 secretion system is there, you can significantly increase the transient expression. And we saw similar thing in Nicotiana benthamiana as well. The transient expression was significantly increased when both type 3 secretion system and the AVR-PTO was present together in the Agrobacterium. Then we looked at the stable transformation. So for this, we used the root tumorigenesis assay in Arabidopsis that was developed in Stan Gelvin's lab. So here we chop the Arabidopsis roots into small pieces and then inoculate with, in this case, tumorigenic Agrobacterium strain that has the PLN18 and AVR-PTO and also the controls without those. And you can see in the last figure to the right, you can see that you can significantly more tumors compared to the controls. And we also did the stable transformation with the non-tumorigenic strain. In this case, we are selecting the root cali against PPT, since we have the PCAS1 binary vector. And again, we saw a significant increase in stable transformation when AVR-PT was delivered through a type 3 secretion system. Surprisingly, we just wanted curiosity, we did the flower dip transformation in Arabidopsis, and we also see a twofold increase in the efficiency in flower dip transformation when AVR-PTO and the type 3 secretion system are present in the Agrobacterium. Then we also wanted to see if other effectors, as I mentioned, we also wanted to see HOP-AO1 and AVR-PTOB, do they also increase our transformation similar to AVR-PTO? And it does. We do see a significant increase in transformation in stable transformation in Arabidopsis for both other effectors, AVR-PTOB and HOP-AO1 effectors. Then we wanted to see if this works in other crop species. So we tried wheat, and in the wheat, we saw that both AVR-PTO and HOP-AO1 and also AVR-PTOB, all the three effectors that we tried, they significantly increased transformation. And also in alfalfa and switchgrass, we tried the AVR-PTO, and it significantly increased transformation. So this clearly suggests that the AVR-PTO is most likely suppressing plant defense response and increasing transformation. So to further prove that it is actually suppressing plant defense response, we looked at the transcripts of several defense response genes. In this case, we used the FRK1, which is known to be a PAM-triggered immunity-induced gene. And this gene is induced upon infection with Agrobacterium, wild-type Agrobacterium. However, when the AVR-PTO is present along with the type 3, you can see on the last column, last bar, it is significantly reduced. The induction is reduced. So this proves that the AVR-PTO can actually suppress plant defense response. And similarly, another defense gene, NHL10, we also see, not dramatic, but at least we see a slight reduction in induction of this gene upon Agrobacterium infection when AVR-PTO and the type 3 secretion system is present. So this further proved that AVR-PTO actually suppresses plant defense response, and that is most likely is the reason we increase transformation efficiency. Then we wanted to see, can we deliver other plant proteins other than other than effectors? Can we deliver some other plant proteins to improve transformation? So for this, we use histone H2A. And a long time ago, when I was doing a PhD in Dr. Stan Gelvin's lab, I had shown that histone H2A plays a positive role in transformation. It's important for transformation. And later on, Dr. Stan Gelvin's lab has shown that not only the full-length HTA, but even a small fragment of the histone H2A is sufficient to increase stable transformation. So we then cloned this histone H2A, and then we fused it with a promoter from the bacterial effector, because the plant promoter will not work here, so we need to use the bacterial promoter, which has the type 3 secretion signal. So we fused this with AVR RPM1, it's one of the effector, and we fused that with N-terminal region so that we have the type 3 secretion signal in the histone H2A. So when we did the experiment in Arabidopsis, again, this is the stable transformation. you can see that the histone H2A and also the truncated histone H2A both significantly increased transformation. Very similar to what was shown earlier. When we overexpress this histone H2A, it can increase transformation in Arabidopsis. And we also tried this in other plants as well. In all the plants that we have tried so far, in Benthamiana, wheat, alfalfa, and switchgrass, the histone H2A again significantly increased transformation. Then we wanted to see, can we deliver, so we showed that we can deliver plant proteins, right? Now we wanted to see, can we deliver other plant morphogenic proteins through this type 3 secretion system, so we could increase regeneration as well instead of just suppressing plant defense response. So in this case, we were using wheat because wheat is very recalcitrant for transformation. So we cloned the homologs of VBM1, WUS, GR5, and GRF4 GIF1 chimera, and also the WOX5. So these are all these have been shown to increase regeneration in other plant species. So we found the wheat homologs of these, and then we fused this with the AVR RPS4 promoter along with N-terminal region, so we have the type 3 signal. And then we transferred this to Agrobacterium, then we did the transformation. This is very, very preliminary data. We still need to repeat this, but the trend looks good. We see that most of these morphogenic proteins can be transferred through Agrobacterium, and we do see increased transgenic plants. We were able to increase transformation using this method. So this was very promising that we could now deliver these morphogenic proteins through this Agrobacterium instead of making a transgenic line having these morphogenic proteins. So just to summarize, so we have shown that the expression of the type 3 secretion system from Pseudomonas is functional in Agrobacterium. And the Agrobacterium is able to secrete effector proteins into the media and translocate into plant cells in the presence of type 3 machinery. And we showed that the Agrobacterium delivering AVR PTO, AVR PTOB, HOP-AA1, and HTA1 can enhance stable transformation in many plant species. And we showed that the plant defense genes are suppressed by bacterially delivered AVR PTO. And the preliminary data suggests that the plant morphogenic proteins can also be delivered through type 3 secretion system to improve plant regeneration. So just ongoing work, we are now trying to deliver multiple proteins such as histone H2A plus AVR PTO, and also the morphogenic proteins plus AVR PTO, so we can suppress plant defense response and also increase regeneration, so that can significantly increase transformation in recalcitrant plant species. And we are also trying to see if we can deliver Cas9 through this type 4 secretion system so that we can achieve transgene-free editing. And Cas9 is a challenging protein to work with because it's pretty big. So we are looking at split Cas9 to see if that can work using this type 3. And also we are looking at some smaller versions of Cas proteins. And some of these are not been shown to work in plants, so we are first trying to see if they can work in plants. If they do, then we are going to use those smaller Cas proteins to deliver through type 3 secretion system to achieve transgene-free editing. With that, I'd like to thank the people who did this work. Most of this work was done by past lab members. Vidya Raman played a major role, and also currently Kashif Mohammed is doing the morphogenic proteins, he's delivering the morphogenic proteins. And the funding from this came from NSF and also initial funding from Nobel Research Institute, and we got a small grant from Bayer as well. So with that, I'll be happy to answer any questions.
We have time for questions.
Yes.
Sujin.
Excellent work. On the last point, can you deliver RNA as well as the protein?
No, right now, yeah. I mean, we cannot do that through Agrobacterium. It has to be delivered through regular tDNA. But we are looking at other ways, for example, viral vectors or other ways that we can achieve that. At this point, we just want to focus on Cas9 first, and then we'll think about RNA next year.
A lot of the morphogenic regulators you looked at are known to increase regeneration in a lot of monocot species, but not so much dicot species. I was wondering if delivery through this secretion system could enhance regeneration in dicots?
Yeah, we have not tested that, but yeah, maybe we need to look at other morphogenic proteins for dicots. But at this point, we were just focusing on wheat. But some of these may work or may not work, but I know that people have tried in poplar and BBM doesn't work in poplar, but yeah, we may have to look at other morphogenic proteins for dicots.
All right, I have a quick question.
Yeah.
So what is the size limitation at this moment, and what are some of the limitations for the translocation?
Yeah, the size, actually, it's a good question because the effectors have been, there are some really big effectors which are like around more than 5,000 amino acids can be transferred. It's been shown, actually, but my guess is that the efficiency will go lower. The bigger the protein, the efficiency will be lower. So that's why we like to try to keep the proteins as low as possible, the smaller as possible. But based on the literature, there are some effectors which are really big, so it can technically transfer bigger proteins, but my guess is that the efficiency will be very low. Yeah.
What's the molecular mechanism of the histone? How it works for the enhancing the transformation efficiency?
Yeah, it's a good question. We don't know the exact mechanism, how the histones work. There's some work has been done through in Stan Gelvin's lab, looking at different stages of plants where the histones play a role, but the exact mechanism, we still don't know how it increases transformation.
Kieran, great talk. So I noticed the expression of the type 3 secretion system machine alone seemed to already suppress the immunity based on FRK1 expression. So what's the explanation, and did you also see the same effect in different strain background?
So you're saying the type 3 secretion system alone without any effector
Yeah,
proteins?
based on one of your results, I saw the decrease of the FRK1 expression. Just the expression of the type 3 secretion system alone, without an effector. Am I right?
As far as I see, maybe there is some error bars, but probably with AVRPTO it is more. But maybe there is a little bit lower. I think maybe it's just a noise, I think. Yeah.
Okay, I just wonder whether the type 3 also delivers some protein from the Agrobacterium into the plant cell to have some suppression.
Yeah, as far as I know, no, it doesn't.
Okay, thank you.
Yeah.
I guess I'm next. Great talk. So my question was like kind of similar to that. So AVRPTO, like as you know, like it decreases the plant immunity expression. And so when you're delivering these, after you're delivering these proteins to the plant cell, does the plant recover plant defense, or does that like immunity like stay suppressed like the entire time? Because I guess my concern is like, you know, when you get these plants to adults, like they still have this very reduced like plant immunity expression. So I'm wondering if AVRPTO is just used just to deliver it and not to permanently suppress it, if that makes sense.
Yeah. So this is the AVRPTO is going to be very transient. It's going to degrade eventually. So the suppression will be very, very transient, only for few hours or few days. So then the AVRPTO will obviously degrade because it's not stably integrated. So yeah, it will not compromise the eventual plant defense response. Yeah.
Thanks.
It's only during transformation.
Hi, Kieran. So in the early part of your talk, you talked about NOG degrading salicylic acid. Can you deliver that via the type 3 secretion system, or are there some size limitations or something to getting it into the plant cell?
Yeah, no, we have not done that, but I don't know how big the protein is. I need to check, but yeah, that's one possibility. Yes, yeah, we can probably deliver that to and decrease plant defense. Yes.
Hi, thank you for great talk. I was wondering if you use the leaves, the monocot leaves, because you're using the morphogenic genes that can increase the regeneration. Have you tried to do the Agrobacterium-mediated transformation in monocot leaves too?
In monocot leaves, no, we have not tried. Yeah, so far we are using wheat. We use immature embryo in wheat. Leaf transformation doesn't work in wheat, as far as I know, but we have not tried other monocots for leaf transformation.
I think we have time for one more question. Let's go. If not, yes. Thank you, Dr. Mysol, for a great presentation.
Thank you.
So Peichen is going to introduce our next speaker for the session.
Good morning, everyone. My name is Peichen Yang. I'm also with Bayer Crop Science, and it's great to see so many experts in the room. It's my great pleasure to introduce our next speaker, Dr. Michelle Valentine. Dr. Michelle Valentine is a native of Missouri. She went to University of Missouri at Columbia for her bachelor and PhD degree in plant science. While she was in graduate school, she did internship with both Pioneer, DuPont, and Monsanto. She completed her PhD with Dr. Zhang Zhen, working on RNAi technology to improve soybean nutrition profile. She joined Bayer Monsanto 7 years ago, and since then she has been working on the intersection of breeding and plant biotechnology. One of the questions we always thinking about, life make rock, and there are lots of recalcitrant lines. It's very difficult to transform. And Dr. Valentine has been working on exploring new technologies and see how we could potentially have new technology to develop tools to transform those lines. Currently, she is on the plant transformation production leadership team, and she's responsible for delivering biotech traits for all the row crops, corn, soy, cotton, canola, for both commercial product development and discovery research. Please join me and welcome Dr. Valentine.
So thank you, Peichen, for the very nice introduction and for the opportunity to speak today on some of our work, which we call treadmill, or the transformation and editing of mixed lines demonstrated here in both corn and soy. And so I'm very happy to share that actually last week, a publication around this work was accepted in the A Biotech Journal. And so it will hopefully be available online or in print, hopefully very, very soon. Okay. So over the last few years at Bayer, we've been working on really strengthening our focus on our mission, which is health for all, hunger for none. And this is the mission of all of Bayer across all the divisions of the company: pharmaceuticals, consumer health, and crop science. For me, working at a really big company, it's really exciting and really inspiring to see that there's so much diversity of really, really good research. But despite all of the diversity of research, it's all pointing at this one mission of health for all, hunger for none. In the crop science division, where I'm at, our part of this mission is to help growers produce enough food at the right time, in the right place, with as few resources as possible. And so this is not only to benefit our customers, the farmers, but also all of us who depend on farmers to make our food. And so in crop science, our vision is to produce more, restore nature, and scale regenerative agriculture. And the first part of that, the idea of producing more, mankind has been doing this, improving our crop plants and our crop animals for thousands of years, with the goal of producing the highest yielding, highest quality plants and animals. But the second parts of that, kind of the newer ideas of restoring nature and scaling regenerative agriculture. At his talk yesterday, Jeff Ahrens did a really nice job of describing our concept around the systems approach of restoring or sustainability and regenerative agriculture, and how we're thinking about that and our actual goals and plans of how we want to get there in the future. And so I think Jeff actually shared this exact slide as well. And so this is a pretty common slide showing that although people have been working on improving crop plants for thousands of years, over the last 100 years, there's been a lot of innovations in agriculture that have been step change innovations to help us use as few resources as possible to grow yield. And so this figure shows, for the example of corn, the amount of land it takes to grow a unit of yield over time. And so looking back about 100 years with the introduction of hybrid corn, my family tells a story a lot about how my great-grandfather was one of the first farmers in our area to champion hybrid corn. At that time, that was a huge innovation, and it was also quite controversial. But going to hybrid corn, that was a huge innovation at the time that helped us produce more with less. And then as we've looked over the last 40 or so years with the introduction of crop protection technologies, transgenics, and then moving into the much more recent years of advanced genetics, precision breeding, high throughput phenotyping, and precision farming, I guess to me the summary that I take away from this slide is that the pace of innovation feels like it's always getting faster. So even looking in the past couple of years with the introduction of short stature crops and especially genome editing, like I said, it's the pace and the scale at which innovation is happening that is so impressive. And so to me, the challenge as somebody who works on plant transformation and genome editing is how can I do my work at the same scale of some of these other innovations that are going around me in agriculture? How can I bring that same kind of pace of innovation to transformation as all of the other technologies so that we can most effectively use all of those technologies together? And so at Bayer, a pretty major update to our transformation system is the use of seed embryo explants, or Cs, for our corn and soy transformation pipelines. And so the exact method of seed embryo explant transformation and the protocols and kind of the science behind it, I believe was described at last year's SAVB meeting, and then it's also in the 2022 publication listed here and a couple of other publications. But the kind of highlight of the protocol is that it begins with dry, mature seeds. And so for both corn and soy, we begin with often field-grown seeds. We use an automated process to extract the embryo explants, and then with traditional transformation and tissue culture, we regenerate fertile, rooted, transgenic plants to hand off to the greenhouse. And so I guess how that relates to my talk is seed embryo explant transformation starts with seeds. And so seeds have the benefit of being able to be mixed. And so the idea of treadmill, or the transformation and editing of mixed lines, is that we upfront, at the beginning of the process, mix seeds from either several germplasm or many germplasm to create a population for transformation. And so then after that upfront mixing, the rest of the transformation process across all the germplasm is done in parallel. And so the idea of treadmill is that we're doing population scale transformation rather than individual line by line. So I guess viewed another way, traditional transformation, at least the way we've traditionally thought about it at Bayer, involves adding our traits or doing our gene edits in either a single germplasm or perhaps a handful of germplasm at the start. And then we test those traits and edits, we look at their efficacy, and then any of the promising traits and edits might get either retransformed into more broad germplasm or more than likely conventionally bred over time into those more elite germplasm. And so that has worked really well for a while, but it does have the limitation of that if you find out very late in this process that your trait or your edit does not work well or does not interact well with the desired germplasm, finding that out very late in the process is very costly. There's a huge amount of time and resources that goes along that funnel of pipeline development, and so finding out late that there are negative germplasm by trait interactions is a huge cost. And so, again, the challenge is how can we more quickly get our traits and our edits into the most relevant germplasm for testing. So treadmill, or the transformation and editing of mixed lines, allows us to generate edits and transgenic events in many germplasm much faster. So again, the overall idea of treadmill is to begin with a thoughtfully created population of germplasm, extract the seed embryo explants from those germplasm, and then in one transformation experiment, all done in parallel under the same conditions, we create transgenic events, and during the normal time where we would already be taking a sample of the transgenic event for things like copy number analysis or molecular analysis, we also take a sample for genotyping. And so then we can decide what genotype each event came from. And so the input of this transformation method is many, many germplasm, and the output is the combination of many germplasm by many traits or edits. And so that helps us do any downstream testing a lot more quickly and a lot more efficiently. You can also think about, there's a lot of ways you could think about using mixed germplasm transformation, but I think another advantage of it is it allows us, as I said, we're mixing the seed from many germplasm up front and doing the rest of the process in parallel. And so, as we know in transformation, there's a lot of experiment-to-experiment variation, a lot of day-to-day variation. And so being able to truly treat all of the germplasm in a population as evenly as possible allows us to more accurately assess the transformability of germplasm. It also is really important for genome editing, where we want to get specific edits in specific germplasm, or perhaps test a variety of edits in a variety of germplasm. I think treadmill is certainly an efficient way of being able to do that. So yeah, I guess all of that to say, treadmill allows us to do transformation and editing at a population scale. So it's allowing us to do what has traditionally been thought of as biotech work at the scale of a modern plant breeding program. And so for the proof of concept experiments that I'll share today, I'll start with the results for soy. So for the proof of concept experiments for the publication, we targeted the determinate 1 or DT1 locus in soy. DT1 is responsible for the the wild type allele of DT1 is responsible for the indeterminate phenotype that you can see in the plants on the far right. So those plants are very viney, very elongated. They continue putting on vegetative growth throughout the reproductive phase. There are natural mutant alleles of DT1 that give a recessive determinate phenotype. And so these plants, at the time where the reproductive phase starts, the vegetative phase stops. And so this results in a much more compact growth habit. And so one of the advantages of DT1 for this project is that it is a very conserved locus. And so trying to design guide RNAs to a multitude of germplasm is a whole lot easier when the locus is more conserved. And so for the transformation experiments in soy, we created a population of 104 elite soy varieties spanning all of the maturity groups grown in the United States. So from 00 down to 7. And so, yeah, we mixed an approximately equal number of seed from each of those 104 varieties up front, extracted the embryo explants, and then transformed them with the construct that's shown there in the middle right. So the transformation construct had the spectinomycin selectable marker, it had the ubiquitin promoter driving Cas12a, and then it had a guide RNA cassette. The guide RNA cassette was targeting the DT1 locus, which encodes the gene TFL1B. And so there were 3 guide RNAs targeting the upstream promoter region of DT1. And so At the end of tissue culture, so like I said, we mixed seed from 104 lines in, we did transformation, and then at the end of the transformation process, we got just over 2,000 events for analysis. And so each of those events was analyzed for a copy number. It was genotyped. We used about 50 TACMan markers to identify what genotype each event came from. So that determined the line ID. And then we sequenced the DT1 or TFL1B locus to figure out what edits were created. And so of the 104 varieties that were put into the mixture, we did recover edited events from 101 different varieties. And so the composition of those events are described here. In the top panel, the different soy lines are on the x-axis, and in this case, they're numbered. We assigned unique number IDs to each line, and they're ordered by their maturity group. So they're clustered by the different maturity groups of soy. And so all of the soy lines are on the x-axis, and then the green bars represent the number of events that were generated for each of those lines or each of those germplasm. And then the blue dots at the top are the percentage of those events that were generated that were also edited. And so as you can see, there is diversity in transformation response among the different germplasm. Some performed better, some performed worse, but we did get events from 101 out of the 104. So while there was diversity in their response to tissue culture and transformation, it doesn't appear to be dependent on maturity group in soy. And then as you can see, the overall editing rate for this experiment was quite high. And so most events had, or most germplasm had over half of their events also be edited. And so for the bottom panel, so while we put an approximately equal number of seed of each line into the mixture, we had different amounts of lines per maturity group. So the panel at the bottom is showing in the green bars the amount of seed from each germplasm maturity group that was put into the bulk. So the green is the input, and then the blue bars represent the proportion of events that were generated after transformation. So the blue is the output. So again, this is kind of another way of looking at the data to show that it looks like the transformation response is not maturity group sensitive. And then looking at the editing results for the events generated. So overall, the editing rate for this experiment was quite high. About 94% of the plants that were generated had at least one edit at the DT1 locus. And so this figure shows the ratio of edited events compared to not edited events for each maturity group. And then the numbers in the middle are the number of events for each maturity group that were generated. So overall, 81 out of the 101 varieties that were transformed had over 90% of their events also be edited. So I think this shows that the system is a very efficient method for generating edited events across a multitude of germplasm in soy. And so looking more closely at the actual sequences, the edit sequences that we recovered. So this data, you might have noticed I said we generated 2,000 events, but then this slide has data from, it's actually almost 1,600 events. So we did get genotyping data from a subset of events and sequencing data from a different subset of events. And so the intersection of those events where we had all of the available data was 1,596 plants. So across the plants where we had all available data, we did get over 800 unique edit sequences at the DT1 site. And so this figure is just showing the top 5 most abundant, or top 5 most abundant edit sequences that we saw in many lines. And so, for example, the 7 base pair deletion shown in blue was seen in events from a majority of the lines. There was a separate slightly different 7 base pair deletion that's shown in red, and it was also seen in many lines. And so overall, across all 3 target sites in the over 1,500 events, we got 800 distinct edit sequences, and we also did see different cutting rates for the 3 different guides. So the lowest cutting guide was about 7%. The highest cutting guide cut in 92% of the plants. And so I think overall, this shows that both if we want to create a single edit in multiple germplasm or a diversity of edits in a diversity of germplasm, it's certainly possible using this mixed germplasm transformation approach. And so that wraps up the results in soy. We also did a set of experiments in corn. For corn, the proof of concept locus was the brown midrib 3 or BM3 locus. BM3 is also a very conserved locus, which is nice. And so BM3 encodes the gene caffeic-o-methyltransferase, or COMT, and knockdown of that gene reduces lignin biosynthesis, which gives the transient brown midrib phenotype, and it also makes the plants more digestible as silage for livestock. And so there are natural mutants and edit-induced mutants of BM3 that have been reported. And so for corn, it was the same basic workflow as we did for soy. For the corn experiments, we made a mixed population of 40 female and 38 male pedigrees that were put into the bulk, so 78 different inbreds. And so by female and male, I mean the 2 different heterotic groups in our breeding program. And again, these spanned all of the relevant maturity groups for North America, from 92 to 117 days. Construct design was also basically the same as soy. We had the selectable marker, promoter driving Cas12a, and then the guide RNA cassette targeting brown midrib 3 gene. In this case, it was targeting the coding region. There were 3 guides around exon 2 of brown midrib 3. So it's important to note that for the corn experiments, we were not able to source exactly equal amounts of seed for each of the lines put into the mixture. We know how many seed of each line was put into the mixture, so we could normalize our data, but it was varying amounts of seed for each of those 78 germplasm. Oops. So again, I guess just to say the same overall workflow of mix the seed, do the transformation, and at the end, sample plants for copy number, genotyping to figure out what genotype went with what event, and then sequencing brown midrib. I think it's also important to note that the corn experiments were done on a smaller scale than soy. So for corn, we generated 139 events total, and those events came, when we looked at the genotyping data, they came from 22 female lines and only 1 of the male pedigrees. And so the top panel here shows the proportion of events generated. So again, that input and output. So the proportion of seed that was put into the bulk mix for each of the maturity groupings compared to the proportion of events that was generated at the end. And so as you can see from here, there does appear to be a bias towards the higher maturity groups for these experiments, but it's important to note that there were more high maturity females put into the bulk than males. And so as I mentioned, there were more female, in these experiments, females outperformed males for transformability. But I will say that we did recover events from both the highest and the lowest maturity groups, indicating that it seems like maturity does not seem to affect transformability in these experiments. But as I mentioned, the females did far outperform the males in these experiments. And so we have data in the publication. I don't have it here today, but we did do a follow-up experiment creating a bulk of only male pedigrees. And so in that follow-up experiment, we sourced seed that was from all grown in the same field location on the same year, and it was much fresher seed. And so for the male experiment, the male-only experiment, we generated events from 9 out of the 36 males tested. So this is not to say that transformation is not possible in males. It was just limited in this experiment. And so the bottom panel, again, shows the proportion of edited events for each of the maturity groupings. Overall, for this experiment, 69% of events had at least one edit at the brown midrib locus. And we did get editing from 17 females, and the one male that was transformed was also edited. So editing in 18 different pedigrees. And so then, again, looking at the actual edit sequences that were created, these again are only the top 5 most abundant edit sequences. We did generate 95 unique edits across the 139 events. And so again, for example, that 3 base pair deletion that's shown in red was seen in 11 out of the 18 germplasm that we worked with. So I think the results combined between corn and soy demonstrate that if we need to create, like here, the same edit across multiple lines or a diversity of edits across a diversity of lines, that is both technically possible. Treadmill is an efficient way of getting there. So to wrap up, treadmill or mixed germplasm transformation has been demonstrated in both soy and corn with the ability to edit and transform multiple genotypes simultaneously. Using treadmill, we can accelerate deployment of our traits and edits into more germplasm more quickly, and that helps us do better testing to find the best traits and edits. And so going back to my original slides on Bayer Crop Science's mission around sustainability and regenerative agriculture, as Jeff mentioned yesterday, we're really thinking about sustainability and regenerative agriculture as a system. We're not just making edits, or just making events, or just making crop protection products. We're creating an entire system and thinking about how all of those things interact not only with each other, but also with the environments they're going to be grown in. And I think it's also a good way of thinking about how we do transformation and how we do gene editing. We're not just making standalone transgenes or standalone edits that have an effect all by themselves. They're really only relevant in the genetic environment that they're being inserted into. And so I think the best way to do testing is to not only test a multiplex of edits, but also a multiplex of germplasm. That'll give the most informative results of how the edits and traits will perform out in the field. And so being able to transform many germplasm in crop plants has been a goal for a long time, and I think treadmill is certainly one way to do that more efficiently. And ultimately, I hope it'll help us make, like I said, more productive, more durable, more sustainable products for agriculture. And so while I had the opportunity and the honor to present today's work, I definitely want to acknowledge all of the co-authors on the manuscript. And then this has been a hugely collaborative effort across both breeding and biotech at Bayer Crop Science. And so I want to thank all of our partners in plant transformation, controlled environment, the genotyping teams, and everybody who helped give both support and advice for this project. And so with that, I'd be happy to take any questions.
A really nice talk. The question I have is, if you produce the same edit in 100 different lines at the same time, how do you manage the workflow in a greenhouse?
That is a great question, and something we are definitely working on. So I am a very small piece of the puzzle in plant transformation. There's obviously a lot of computational work that needs to go on upstream, a lot of greenhouse and field work that needs to go on downstream. And so, yeah, I guess for SIVB, we talked about the transformation side of it, but we are definitely looking into how do we manage and efficiently work in multiple germplasm in all areas of the pipeline.
Hi, Michelle, thank you. That was really interesting approach looking at different genotypes. I was curious, how many, for the soy, how many seeds did you use per, and how did you determine that number?
So I think we do say in the manuscript we used about 5,000 seed from each of those lines. And so that is something I recognize working at a seed breeding company. I have the luxury of access to a lot of seed, so that is a consideration. I'm sorry, what was this second? Oh, so I guess we do have predictive models that help us determine if we want a certain number of events at the end, how much inputs we should do. We definitely did work with statisticians to make sure that we were getting enough plants to be able to make calls, especially for the manuscript.
So you said you tried to use this kind of similar maturity group of the germlines or the lines. But even though that I can see some, like the 2 weeks of the difference in the maturation dates, and how did you synchronize to initiate the experimental plant transformation, like within these 2 weeks?
So the maturity groups are, if you were to plant those seed in the field, about how long it would take for them to reach maturity. And so we, especially in these experiments, did not see a maturity interaction with our tissue culture system.
Nice talk. As you showed your workflow, so you are screening upfront for the copy number or quality. And then, but I believe you could do so because you have the data for that. And so I wonder if you could share some information about, was there some association with the maturity group and copy number or position effect of the tDNA insertion or something on those lines? Whatever you could share, if there is some information you were able to gather.
So I'd have to, I guess there was a lot of people who worked on this, and we looked at the data in a lot of different slices. And so I do know we looked at especially editing rate by certain genotypes. I don't know if we looked at, I know we looked at things like copy number by genotype, but I don't know if we published it. But I think that is a benefit of transforming multiple germplasm at once, is you can start to make some of those more fine-tuned assessments of how germplasm interacts with those factors.
Yeah, thank you very much. It's really nice talk. And how do you determine the important regulatory element in the promoter region to edit? Which criteria to use? And when you use multiple guide RNA to edit the target regions, would it be increase the chance of off-target activity? So if can increase the chance of off-target activity when we use multiple guide RNA, what is the best way to minimize the chance of off-target activity when we use multiple guide RNA to edit the target regions, please?
Okay. So as I mentioned, I'm in transformation, and so the exact design of the guide RNAs and kind of the rationale of why putting the guide RNAs in the regulatory region of DT1 sounds like an easy answer, but that's a different team. And so we were just happy to use guide RNAs that were conserved across our lines and we knew had good editing efficiency. We do, when we design guide RNAs, we do want to make sure that we minimize any potential off targets. And so I know a lot of that depends on knowing the sequence of the germplasm you're working on. So that's definitely another consideration. But we try to minimize off targets computationally.
Now that you have transformation efficiency information for all of your lines, would you alter the relative amount of seeds you'll put in for these lines so you get out a more even number of transformants?
That's a great idea. That would be something you could definitely imagine doing, yes.
Hi, nice talk. Two things. First is that I think Corteva really needs to up its acronym game.
Yeah.
We're falling. And the other, the question that I have is, seed source, seed quality can vary tremendously. And do you think some of the variability you're seeing might just be because you don't have consistent seed sources?
Absolutely. So as I mentioned, the original corn experiments sourced different seed from different fields, different growing locations. We had an idea that we wanted to test, and so we tried to move as fast as possible using the seed that was available. I wish I would have put the figure in this presentation, but the follow-up male-only corn experiment was seed grown in the same field location, the same season. It was much more recent or fresher seed, and I do think we got better quality data from that. And so that is absolutely a caveat to this data in that the germplasm effects we see could also be seed effects, seed quality effects.
Hi, I want to continue Todd's question, and we also noticed that when we're using tomato, other seeds growing in a different environment, even in the same location and different seasons, we found the regeneration and the transformation efficiency is very, very different. Have you considered any treatment before you using the mature seeds for infection?
Yes, making our system the most reproducible and the best efficiency is obviously in our best interest. And so, yeah, we are working on trying to create the highest quality seed for explants. I don't have any specifics of what we're thinking of, but if you have ideas, that'd be great.
Any more questions?
So let's give another round of applause to Dr. Valentine. Thank you.
Good morning, everyone. So my name is Alfred Ho, and from University of Florida. So, okay, all right. So our last speaker for this session is Dr. Sungba Lee. This is my great pleasure to introduce Dr. Lee and also my friend and also excellent colleague from Iowa State University. Dr. Lee is currently a research scientist and adjunct assistant professor from the Department of Agronomy in the Iowa State University. Dr. Lee's research focuses on the development and improvement plant biotechnology tools for crop genetic transformation and genome engineering. Dr. Lee's group aims to broaden the spectrum of transformable crops by engineering agrobacteria strains. I think I want to get one from his lab, and also use new technologies for the genotype flexible plant transformation. Welcome, Dr. Lee.
Good morning, everyone. I first want to thank organizers for inviting me to give this presentation. Actually, I was a little bit conceived by Deepika, because when she first invited me, she didn't tell me about the session. She didn't give me how long will be the presentation. So I thought, okay, maybe it'll be a 15-minute talk. But once I accepted it, she sent me more details. It's like 35 minutes. Okay. So I added some more details in addition to the non-integrating Wushel mediated gene editing. So bear with me. So I'll give you some introduction material about ternary vector system for plant agrobacterium mediated plant transformation. So this is the outline. First, I want to emphasize again the importance of a ternary vector system. If you are experiencing difficulties delivering the transgene into the plant cells, especially monocots, this ternary helper plasmid will be really beneficial. And then I will talk about our rapid maize B104 immature embryo transformation. This is not really our own invention. We implemented Corteva's Quickon media regime. We discovered that without DAB genes, we were able to regenerate the B104 plants very quickly within 7-8 weeks. So we published the method paper. And the last part is about using non-integrating Wushel or altruistic transformation. We tested it for P73 transformation, and we were able to recover some transgene-free edited lines. So Kieran gave a really nice introduction to Agrobacterium. So I'm just focusing on the different vector systems. So originally, the Agrobacterium strain, as a natural genetic engineer, it's unitary vector. It has most of them, not all of them. Some agro strain have multiple TI or RI plasmid, but in general, they have a tumor-inducing plasmid, and it has the DNA delivery machinery, which is a type 4 secretion system, and then the natural tDNA is also encoded on the same plasmid. So this is considered the unitary vector system. After discovery of the border sequences and that it's separable, dispensable, then the binary vector system was developed. Because the unitary vector system, the co-integration, meaning that you clone your gene of interest into a vector, and then you have to introduce an agro there. Your gene of interest must integrate between the two border sequences on the TI plasmid. So that process is really tedious and less efficient. Therefore, the development of the binary vector system really boosted the plant transformation, and it became really popular. So the derivative of the binary vector system is a super binary. I'll describe a little bit more detail next slide. And then came the ternary vector system. So the earlier version was using just a VRG, constitutively active VRG on a separate plasmid that enhanced plant transformation. However, the current, more advanced version was developed by Corteva in 2018 by putting a complete functional set of virulence genes on a compatible plasmid. So now there are 3 plasmids. Disarm the TI plasmid, your tDNA vector, plus the accessory helper plasmid. So there are 3 plasmids. Maybe you can add a 4th plasmid and then 5th plasmid in the future, right? Let's see what's coming. So again, I already described it. Binary vector system is popular because the cloning is really simplified. We can easily modify because it is smaller. Therefore, the pigrin-based vectors, they wanted to make it even smaller. Therefore, they removed all the backbone, only providing the necessary replication initiation factors on a separate plasmid, like a pigrin pea soup. So I don't know if we can call it ternary vector, but still, the intention was to make the cloning more simple. So then came the super binary vector system developed by Japan Tobacco, the famous scientist Dr. Komari. The Komari fragment, because at the time the sequencing was not easy. So usually after digestion, this is KPN1 fragment cloned into a vector, binary vector. Therefore, you are having extra virulence gene, and that really allowed the transformation of recalcitrant monocots. That really changed the field of monocot transformation, especially maize. The difficulty is that just like a unitary vector system, the cloning step was very, very tedious. You have to co-integrate it. Not many people can easily do that. Therefore, it was a really, really slow down. So ternary vector system, by separating the super-binary into 2 pieces, right? One is a tDNA vector, so just like a regular binary vector, but having an assistant or helper plasmid with a compatible original replications. Then you can really boost the transformation efficiency. So therefore, you combine the benefit of a super binary vector and then easy cloning of the regular binary vector system. You can take advantage of this ternary vector system. So this is our older version of ternary vectors. We still have a disarmed TI plasmid and a tDNA vector. As a helper, I made this PKL2299. All the virulence genes are from the hypervirulent BO542 TI plasmid. And then I put it on the RK2 original replication because it's compatible with many existing tDNA binary vector. So I did not want to change the whole things. I just want to utilize our existing vector system that has been working very well for maize transformation and soybean transformation. So therefore, I looked up Corteva's paper and found that this original replication is compatible with many existing binary vector systems. So whoever interested, just check it compatibility, including original replication and also antibiotic resistance gene. If they are compatible, you can put them together. For tDNA construct, for this presentation, we use the two constructs. One is the CRISPR-Cas9 construct, so PKL2359. It has a mCherry, RRP reporter, and then maize codon optimized Cas9 targeting GLOSI2 gene. So GLOSI2 gene is responsible for the deposition of a cuticle layer in the juvenile tissue. So the loss of function mutant, it can easily screen by spraying water. So its mutant will have a water adhesion, but the wild type will just let the water drain because it has a wax layer. The next vector is a reporter construct. Now many of you already heard about it. It's a Ruby construct. When you first heard about it, wow, you don't need to sacrifice your plant tissues, right? You can easily visualize it. You don't need a microscope, no confocal microscope. You don't have to open your culture plates that can risk the contamination. So you can easily visualize it. So we immediately implemented it and then and test it for transformation. So I will just simply repeat. So Ruby convert tyrosine into a purple pigment. Dr. Zhao's lab made this marker by combining 3 key enzymes that converts tyrosine into purple pigment betaline. And those 3 enzymes are under control of one single promoter and terminator. And then after transcription and translation, they will be separated by the self-cleaving peptide 2A. So this way, you don't have to worry about the expression of 3 different enzymes at the same time. So now this is a quick summary of a rapid B104 transformation method. Once again, this is not our invention, but we are utilizing Corteva's quick media regime. At day 1, you isolate immature embryo and do the infection for overnight. So next day, we move them to the resting media, no selection, just resting for a week. And then we move them to the maturation media, maturation and shooting with the selection. It takes about 4 weeks. You already see the regenerating shoots at this time. And then you move them to the rooting for 2 weeks. You already get rooted transgenic plants. This image just shows that how you isolate immature embryo. I don't have to go to detail, but this is a really tedious process. And I'm glad that I don't do this myself. I have a team, and Minjung Kang or MJ, she has done a lot of this work. And then now ISU's crop bioengineering laboratory, Dr. Jessica G is here. She is doing all the transformation for me, and I'm really thankful. So here is some images. During resting period, you can check the transgene expression. With mCherry, you need a fluorescent microscopy to take the images. But with Ruby, you can immediately see with the naked eyes. You don't even have to open your petri dishes, right? Then there's no contamination issues. And then you put them into maturation, then you can see the development of the shoots. And when we use the Ruby, you can also see, you can easily see the expression of Ruby and pigmentation. And then we move them to the resting, and then after 2 weeks, you can get the transgenics. So now it's time to move from in vitro to in vivo. So I'm not going over detail, but once you get those rooted plant leads, take good care. And if you're interested in generating T1C, then you move them to the greenhouse. So we found that Ruby is really, really excellent visible marker. So we started to use it for other transformation work. So in immature embryo, callus, regenerating shoots, or seedlings and roots, you can visualize the pigment in all the tissue types. So this is really great work by MJ. She took all the images, nice images, and make the cross sections of root and shoot and stem. And we couldn't see the pigment in the pollen. Transgenic pollen is yellow, so you cannot tell the difference. But anthers are really colorful. Carp, even the seed, if you cut the seed, you can see the pigmentation there. And what's fascinating is that if you look at the 4 ears at the bottom, you can see the variation, right? The second one, you can see the one-to-one segregation of Ruby. So if you are interested in using Ruby to track the transgene, then I think this will be really, really good. But you would want to select a really good promoter. Here we use a cauliflower mosaic virus promoter. It looks like it's quite strong. So as you can see on the plant, the 3 plant and panel A, we move them to the soil on the same day. However, their growth and development is totally different. When the plant exhibit or accumulate too much betalain, meaning that they have a tyrosine depletion symptom. So they do not grow normally. Therefore, there's a room for improvement. Maybe you use a slightly weaker, much weaker promoter for constitutive expression, or tissue type specific promoters if you are interested in the tracking transgene segregation. So we also had this quick quantification method for betalain. So in case you're interested in transgene expression analysis, like you may use this one to optimize some promoters, terminators. The quantification method is really, really reliable. Compared to Gus staining, this will be really, really much easier. It'll save you a lot of time. Okay, Vijay is a poster. His poster is a 2088. And then on some of these vectors available, we have a newer version of a helper plasmid, PKL2299A. I really made it because of Stan. One day, where's Stan? Is here. He asked me one day, do you have a helper plasmid with A? At the time, I did not have one. So I created one and tested it, and it enhances transformation. So that will be available very soon. And if you want to test the Ruby construct, we have a PCBL101 Ruby. Sorry, there is a mistake. So CBL101 Ruby has MPT2 for selection, but CBL102 Ruby has HRA for plant selection. Okay, so let me repeat this a little bit. Our bottleneck in plant transformation still is genotype dependence, and then X plant dependent, and the low transmission efficiency. We always want to break this bottleneck. We heard about different approaches by Kieran's engineering Agrobacterium strain, right, delivering effector proteins to soften the host defense, or using morphogenic transcription factors. So especially baby boom Wushel technology has been proven really efficient for Manaka system. And if you look at here, this shows the gene regulator network for shoot apical meristem. These results are mainly based on the model system, including Arabidopsis. And then the bold, the morphogenic transcription factors with the bold indicate that they have been tested at least somehow, right? Whether they are practically useful for your plan of interest, we do not know. There's no guarantee. However, they have been tested that they somehow enhances regeneration or plant transformation. So what I want to point out here is that look at the Wushel, right? Wushel is the central player to maintain the shoot apical meristem cell at the organizing center. and then it's a key player for the stem cell populations. So the key feature of a Wuschel is that it diffuses, and then this Wuschel gradient is the key to generate these stem cell populations. So I don't know how Bill came up with the combining baby boom and Wuschel. By looking at this network, I have no clue, which one you have to choose and how you combine it. We know that IPT is good for some dicots. When you combine IPT and Wuschel, it has been very successful for dicots. For monocots, baby boom and Wuschel. And then Corteva's new technology using LAC2 and baby boom is very successful for dicots. I cannot tell by looking at these regulatory networks, right? So there's a lot more to do for us. And then this is just a table just to summarize some of the recent publications. They tested the different morphogenic regulators, and then some of them go through induces organogenesis. Some of them induces somatic embryogenesis or organogenesis. But you cannot tell by looking at the literature whether these specific MTF is useful for your own plant species or genotypes. So, as I said, the baby boom Wuschel works best for monocots so far. So therefore, we have been interested in using baby boom Wuschel. But we all know that constitutive overexpression of them will not allow the somatic embryos to regenerate into normal plants. Therefore, different approaches has been developed. As you see here, A is an inducible excision, right? You let the baby boom Wuschel induce somatic embryos. After the later stages, you excise them out using Cree recombinase. Another approach is using inducible promoter and tissue-specific promoter combination. However, this approach will be limited to certain tissue types or cell types that allow the sufficient expression of both baby boom and Wuschel. And the C is most interesting to me because it's easily adaptable, right? You can just combine with your existing vector system. As I said, I chose our ternary vector for that. I want to use something that is easy to implement. So non-integrated Wuschel, you just need to have an additional Agrobacterium strain and an additional which carries a different tDNA vector. In this case, Wuschel expressing cassette, very strong promoter such as a PLTP. Corteva tested PLTP also with or without the viral enhancers, which further enhances gene expression. And these days, many labs have been testing different morphogenic genes, such as GRF4 and GIF1 and GRF5, maize WOX5, wheat WOX5, and PLT5, Politora5, and one is not included here is a Wisconsin group, Frank WOX2A, maize WOX2A, is also known to enhance the regeneration in recalcitrant B73. Another approach is developed by Yiping's group using CRISPR combo. This is based on the observation that short guide RNAs like 15 nucleotide, even if you are using CRISPR nuclease, it will not generate double-stranded break. So previously, the CRISPR A or CRISPR I utilized the dead Cas9 because you don't want to introduce any indel mutations. But if you're using small guide RNAs, you can avoid that cleavage. Therefore, if you combine the two guide RNAs, so combine the system using Cas9 nuclease, you can achieve a gene activation at the same time targeted gene editing. So why we call it non-integrated Wuschel? Because there's two scenarios. First is maybe some cells have a stable integrated Wuschel cassette. So Wuschel is expressed and diffused to the neighboring cell and stimulating cell division. Another scenario is that because you are mixing usually one altruistic strain and 9 of your gene of interest agro strain, so there's one red agro there which delivers Wuschel, but most agro cells will deliver your gene of interest tDNA. The outcome is not all tDNAs are integrated into the chromosome. So therefore, sometimes you get transient expression of Wushel and without integration, that is just sufficient to stimulate morphogenesis. So therefore, as I said, NIW approach does have advantages. Okay, you don't have to make the complicated vectors. You can just use your existing binary vector system or ternary vector system. You just add one more Agrobacterium strain, which carries this Wushel expression cassette. So for us, as I said, we liked Ruby. We wanted to monitor where the Wushel cassette landed. So therefore, we combined the Ruby cassette with the Wushel cassette. So we used the very strong mage Wushel promoter combined with Corteva's the triple Bayer enhancer. That's a NIW strain, but at the same time, we want to test how different Agrobacterium strain behave, or whether our new thymidine oxospecific strain, such as EHA105, or the Thai-minus, Rege-minus strain, EHA105TR, can be used for this type of applications. For our gene of interest, we use the LBF44 Thai-minus, and then the tDNA vector was a CRISPR-Cas9 construct. So this is the procedure. You grow agro as you always do, but before the infection, you just mix with a 9:1 ratio. We do co-infection. The procedure is the same. 1 day of co-cultivation, and we move them to the maturation and regeneration. So the Wushel integration occur, and then it will diffuse to the neighboring cell. So hopefully, we can identify the transgenic material, because the Wushel cassette, we did not put any selectable marker gene. So they will not survive anyway. So this is a summary of the B104 transformation. First, we want to see, with the NIW approach, can we enhance B104 transformation frequency? And then here we tested, the control means that there's no NIW strain, but we tested 3 different Agrobacterium strain, LBA44, EH105 Thai-minus, and EH105TR. We used a similar number of immature embryos, and then we were able to regenerate not too many, but with a good transformation frequency, about 10% without control. And when we mixed with the same strain for NIW, we almost doubled the transformation frequency to about 19%. And we rarely saw co-integration, because you can visually monitor the integration of the Wushel cassette. But we all know that B104 is transformable. That's not quite exciting. And the question is, can you transform the reference genotype B73? So for those who are not familiar with the maize, B73 is reference genome, which is the first sequenced inbred line, and it was developed by ISU, but it has been widely used for other QTL mapping or breeding programs. There was a huge demand for B73 transformation, but it was not transformable using, without the conventional method. Therefore, its close relative B104 became popular because it's highly similar to B73, but is transformable using conventional method. So our question was, can we transform B73 using this NIW method? And the second part is, because B73 are not regenerable without the NIW, can we regenerate transgene-free edited lines? So the approach is the same. The only difference that we did not add any selection reagent during the entire tissue culture process. And we assumed that there are 4 types of scenarios. The blue ones, you have a gene of interest integration so that it can survive the selection. Second is Wushel is integrated. We color the pink because it has a Ruby marker. Third one is it has only transient expression of Cas9 tDNA, therefore it can have edit. Normally it cannot regenerate, but with the help of Wushel, it can regenerate. And then the fourth case is just wild type cell. no integration at all, but they were just sitting next to the Wushel integrated cells, therefore they can regenerate. But the Wushel integrated cell cannot regenerate, therefore we will obtain 3 types of plants, and then we screen them, and you will see that numbers are too small. Sorry, we don't have the capacity to grow thousands of embryos. So we will only do this proof of concept type of study. And then our control, we use 136 immature embryo. As you can see, nothing regenerated. With the NIW approach, when we use the same LBA44 4' strain to deliver both Cas9 construct and the Wushel construct, we infected 395 embryos and regenerated 79 plants. That's exactly 20%. 20% of embryos were able to regenerate some shoots. And then we did a PCR screening to identify transgenic lines. And then we identified 9 of them. So in terms of the transmission frequency, that's equivalent to 2.3%. It's okay. I hope it doesn't sound too bad. Yeah, and we didn't see any co-integration event. And then remaining 30 plants, we did a PCR screening for the target gene, and also did the check the presence of the other part of the tDNA or vector backbone. So at the bottom, you can see that some of them already have a M-cherry expression, which is included in the Cas9 construct. So therefore, you can identify some transgenic event. And the loss of function mutant, like a biallelic mutant, when you spray the water, you can tell the phenotype. So we have observed some loss of function mutant. But importantly, among the 70 wild-type plants, we discovered 3 of them were free of transgene. So for those 3 lines, we designed the extra, with a total of 7 primer pairs to check the presence of the tDNA or backbone part, but we were not able to detect any of them. So we think even if it's not whole genome sequencing, from our end, we think it's transgene free. However, of course, if you go deeper, and you may find some pieces of DNA there, but because we did using Agrobacterium, we hope this is a cleaner event. And one event has a biallelic mutations, 4 base pair deletion and 27 base pair deletion. The other 2 plants have only 1 allele was mutated. So based on these observations, we think transgene-free editing is possible for some P73. And this is a transgenic P73. Of course, it's nothing new. If you deliver tDNA to P73, you will recover edited lines. So to summarize it, we tested a non-integrated Wushel approach for maize transformation. We saw that it enhances the B104 transformation by about 2-fold. If you optimize it, you can further enhance it. And then we found that it is useful for recalcitrant major transformation, such as the B73, especially for transgene-free editing. I don't think a B104 is a good option because you will end up having hundreds of regenerative plants and you have to screen all of them. But recalcitrant genotypes, they usually don't regenerate. I think there's a better chance to recover transgene-free edited lines. And I'd like to thank my supervisor, Dr. Kan Wang, who has been very supportive. And then here I highlighted MJ Kang, who did most of this work. And then my collaborators from Crop Engineering Laboratory, Dr. Jessica Ji and Sehija, they have been really, really supportive for transformation. And then Dr. Eric Fulbrick, who is a major geneticist from Iowa State University, who provided the B104 and B73s. And most of all, Dr. Bill Goldenkamp from Corteva for your generous sharing your material and construct and giving us advice all the time. We're highly thankful. And Vina is our collaborator for the rapid B104 transformation method. So we work together and we demonstrated that the rapid transformation of B104 is possible without the gene. It was demonstrated at both ISV and the Danforth Center. And for fundings, I like to thank ISU's Crop Bioengineering Center for the seed grant and the NSF and NSF App Score. Lastly, this is a picture of my group. Frontline, you can see Dr. Kan Wang, and then MJ recently graduated, and Jacob also graduated, and then we also have 3 new members. So I'd like to thank all of them, and thank you all for your attention. I'd be happy to take any questions.
Thank you so much for your presentation. If I well understood, in one of your sections, you mentioned how this gene Ruby can be used for visual phenotyping. And from what I understand, you say that there is a fitness cost associated with using it. Have you maybe tried supplementing either the media or the plant itself with tyrosine or some other fertilizer?
Yeah, that's a good question. Yes. It can work to some extent, but we don't want to make things complicated, as simple as possible. Yeah, for some T1 seed with a high deep color, they have difficulty germinating. But if you soak in the tyrosine, they can germinate better. Yeah, you're welcome.
Thank you so much, Dr. Lee. It was a great presentation. I have one comment and 2 questions. One comment is that you see that the Ruby is not expressed in the pollen, but it is, as I know, it's 35S was not working in the pollen. That might be the reason you cannot observe that the Ruby in there. But I think it might be the good approach is eventually because that is not affect to the pollen viabilities in case, right? And one question is that when you tested the ternary system, I want the increase the transformation efficiency you mentioned. I wonder how much is increased compared to that the binary system. And second one, you mentioned earlier in the workshop that there is a trade-off when transformation is increased, but the copy number is increased too, right? So I wonder is that do you have some kind of a metric or numbers, how much rate of the multi-copy numbers when you use a ternary system?
Yeah, thanks for the question. So first of all, sorry, I've already forgot the first question. Yeah, transformation efficiency. We recently tested it for thiamidine oxotropic EHA105 strain. We were surprised. We were using this new QuickCom method, which has a very short co-cultivation period. It's about 20 hours. We were not able to recover any transgenic without the ternary helper. So with the new ternary helper, when immature embryo quality is good, we can get almost all embryo regenerate. So because Jessica is so good, she makes so many transgenics. And the second question about whether the ternary helper plasmid increases a copy number, based on the literature, that is yes. Have we tested? No, because for us, without ternary helper, we have no option, right? We have to modify. We have to go back to our old protocol, which has a longer cultivation period, and then maybe change the Agrobacterium cell density. But we want to keep the protocol as simple as possible. So. We don't have a real direct comparison.
Very nice work, Gunsub. I have a question about betalain. Do you have any idea about the stability of betalain, the red color, if the gene is not there? Okay, so if you don't have the Ruby gene,
so yeah, we have done that's a good question. Whether it as a marker gene, right? We did a lot of recently did a lot of agro infiltration with the Bentamiana, and then betalain did not disappear over time. You can actually have a higher accumulation, and they remain there.
So okay, so do you know whether those cells were transformed stably or not, and how long can you keep it around?
I don't remember the exact day, but we used it for workshop, and then maybe 2 weeks later we extracted betalain for quantification training. and they were really, really purple. So I don't know if the transgenes are still there or not, but at least, looks like the bethylene itself is very stable. Agro-infiltration.
But that delivers some integration of the genome.
They can be integrated, yes. But we have not tested whether they have a stable integration or not.
Hey, Kenso, great presentation. So you mentioned, at the beginning, the super binary vector, the problem was cloning was difficult. I was wondering if the ternary vector system, could you use the super binary vector and just add an extra ternary plasmid with the virulence genes?
Well, that is, I don't know, is there anyone who would want to test it? You mean extra copy?
Yeah,
now
because then you have three.
Yeah. Now I am testing multiple vectors.
Yeah.
There is an option for me to add extra
Another.
virulence genes, but the thing is, there will be some cost there.
Okay.
Yeah.
Thank you.
I want to bring it back to the first talk. Are you considering making a helper plasmid with type 4 circulation system? Would that
You mean
work?
type 3?
Type 3, yeah, sorry. Misspoke.
Well, Kiran's here, right? I would not make any conflict.
Any more questions? So any question for our other speakers? Deepika, you want to wrap up this session? Oh, there's one more question.
So you use a thiamine minus and rec A minus for the transformation. I wonder how important are those mutants in terms of transformation?
Yeah, that's a good question. I don't know, when we used the EH105 prototype strains, we did not have much overgrowth issues. Usually it's AGL1, which gives you a nightmare overgrowth issues. But we know that using thiamine and oxotropic strains, you don't have to worry about carryover because they cannot survive without supplementing thymidine. So it's a kind of a precaution. Even if with our explant material, we don't have overgrowth issues as a major problem. But we know that if you use the thiamine oxotropic strains, the strains have less likely to survive outside of the laboratory conditions. So especially using CRISPR reagent, we want to add another layer of security. So we are hoping that can help for that. Not necessarily overgrowth issues per se. We have not made a direct comparison, but when we made the thymidine oxotropic AGL1 strain using Stan's strain, we saw some decrease. Looks like the thymidine oxotropic strain is slightly weaker in terms of DNA delivery. However, we had to lose some embryos because of overgrowth. So at the end, it became, the outcome was very similar. At the beginning, we had more immature embryo with the Ruby expression with the prototype AGL1, but we lost some of them because of the overgrowth. So there is some trade-off there. But we don't know, to what extent the thymidine oxotrope suffer the fitness.
Any other questions?
Thank you.